Okay, hi everybody. Welcome back to the third part of our photosynthesis notes. We're going to talk about the light-independent reactions. You should have watched the intro to photosynthesis. You should have watched the light-dependent reactions videos first, and now we're going to wrap up with the light-independent reactions. They are not called dark reactions, by the way. They are called light independent. It's also known as the Calvin cycle or carbon fixation. Don't you love it when scientists have all kinds of words for the same exact thing? Super annoying. All right. It's named after the scientist, the American scientist, Melvin Calvin. I love that name, who won the Nobel Prize in 1961. I mention it because this is the kind of thing that AP, the College Board, includes on their AP exams all the time. It's not that you need to memorize it. It's that they're going to tell you about his experiment and expect you to understand how it worked. So he added radioactive carbon-14 to photosynthesizing algae. And then after specific intervals of time, he killed the algae and studied where the radioactive carbon was in the carbon compounds. So which carbon compounds had been formed at different times during the cycle by locating the radioactive carbon-14. And then he was able to figure out exactly what's happening during the carbon cycle, or during the Calvin cycle, sorry. And then he won the Nobel Prize for that. And I'm not sure why it's not moving. I don't know what I did wrong. There we go. Okay. So here's an overview. Let's just listen to the overview first, and then you can decide what to write down. In this case, the diagram on the right might be the best thing instead of writing all the words. And the diagram on the right, as we finish through the notes, right now part of it is grayed out. It will not be all grayed out. Once we get to the end of the notes. Okay, so the overview. These are reactions that are occurring in the stroma. The light-dependent were occurring in the thylakoid membrane. These are occurring in the stroma of the chloroplast itself. There are three main steps. The first step, look at the diagram, see if you can identify them. The first step is called carbon fixation. Notice that carbon dioxide molecules are being removed from the atmosphere, and they're being attached together into a molecule of what's called GP, glyceraldehyde phosphate. You don't need to know that, but that's just what's happening. The carbon is being attached. Remember, we're trying to grow a tree from gas in the air. All right, the next stage of events, and there are like 100 different reactions that happen during the process of, I probably exaggerated, but anyway, a lot of different processes that happen during Calvin cycle. We're just summarizing them. The first one we're going to call carbon fixation. The second one we're going to call reduction. What does reduction mean again? Oh, right, the gaining of electrons. So remember that we've grabbed electrons from water and we're adding them on to molecules here to build our carbohydrates. And then the last part is called regeneration. We're going to remake some of the molecules that start the whole process so that the process is cyclic, so it can keep happening. During the Calvin cycle, ATP, which we just made a few minutes ago, and I guess that was on my previous video. To me, it was a few minutes ago, and NADPH get used up in this process. The ATP is powering the process, and the NADPH is dropping off its hydrogens and its electrons. A 3-carbon molecule is produced with each rotation, sort of. It's not rotating, you guys, but in a diagram of it, it's rotating. So with each turn of the Calvin cycle, we make a 3-carbon molecule, and then 2 turns of it, those 3-carbon molecules get put together to make something like glucose. So you really need to pay attention to the number of carbons that are involved. So for example, 1 carbon dioxide just has 1 carbon. But if you take 3 carbon dioxides in, now you have 3 carbons. So every single turn of the Calvin cycle, you're adding 3 carbons. So if it turns twice, now you have 6 carbons, and that's the number of carbons that are in a molecule like glucose. Okay, so let's go into a little bit more detail. So step 1, carbon fixation. The cycle begins, remember we had to do regeneration, so there's already a molecule that exists. It's a 5-carbon molecule called ribulose bisphosphate. Find it on the diagram there. Do you see where it says RUBP? That's a 5-carbon molecule. We're going to add carbon dioxide to it, which makes it a 6-carbon molecule. There's an enzyme called rubisco, super important, the most important enzyme on the planet. It was what allows life to exist on this planet, this enzyme called rubisco. It's the one that attaches the carbon dioxide molecule to the RUBP. I would add that label to if you're going to just diagram this diagram, I would add the label rubisco and show that it's the one that's attaching the carbon to the RUBP. So it's making it a 6-carbon molecule. The resulting 6-carbon molecule immediately falls apart into 2 3-carbon molecules. So those 3-carbon molecules are called glyceraldehyde phosphate, or G3P, and locate the GP on the diagram there. So it immediately makes these 3-carbon molecules. One cycle, so one time through the Calvin cycle, involves 3 molecules of ribulose bisphosphate combining with 3 molecules of carbon dioxide, creating 3 molecules of glyceraldehyde phosphate. Do you need to know all those long words? Absolutely not. So just pay attention to the number of carbons and you're doing great. All right, so step 2 is called reduction. And now my picture is in the way of what's happening, so I'm going to bring it up there. Okay, remember that reduction means the addition of electrons and that hydrogen ions also often follow. NADPH that we made in the light-dependent reactions, remember they caught the electrons, they were electron carriers. They bring the electrons over to the Calvin cycle, and it says NADPH transfers electrons and hydrogen ions to that 3-carbon molecule. So it drops them off, and now it becomes NADP plus again, and that NADP plus then returns to the light-dependent cycle to gain its hydrogens and electrons again. So it's constantly cycling. We also use, or the plant also uses ATP at this point. Energy is released when the 3rd phosphate is removed, and that powers some of these chemical reactions that are occurring. Again, this process is way more complicated than we make it look, and I'm sure you're like, it can't be any more complicated than this. Anyway, energy is released, so now we have ADP and inorganic phosphate. That's going to get put back together again over in the light-dependent reactions. One of the 3-carbon molecules is then released from the process so that it doesn't continue going through the cycle, and the others continue on through the cycle. The 3-carbon molecule that leaves is then bonded to another 3-carbon molecule to form the 6-carbon glucose or other carbs. In the last stage, step 3, it's called regeneration of RUBP. The remaining, remember we had a whole bunch of 3-carbon molecules, they get put back together to make 3 5-carbon molecules. So let me back up. You had, don't worry about these numbers, I'm just helping you figure it out. You originally had 18 3-carbon molecules. We lost one of them. It's going to go build a glucose molecule. So now you're left with 15 3-carbon molecules. They get all rearranged, and this is during this regeneration process, they get rearranged to make 3 5-carbon molecules. How many carbons do we have now? 15. So if you do the math, it still works out. So the remaining 3-carbon molecules are rearranged to recreate the 3 5-carbon RUBP that started the process. ATP is needed to power that regeneration, so we're going to use up some more of our ATP in the process. All right, that's it. We know all about photosynthesis. Let me just review a few things at the end here to make sure that we're really clear. So this question says, can you answer these most basic questions? I got super complicated in photosynthesis, but can you keep your eye on the prize, you guys? Can you remember what's actually happening here? Where does the dry mass of a tree come from? One of the largest of the redwood trees known, I forget what that says there, hold on, the Lindsay Creek redwood, was estimated to weigh over 4 million pounds, about the weight of a Saturn V rocket. If it started as just a seed, where did all the mass come from? And hopefully you're thinking to yourself, well, KJ, I know the answer to this. It pulled carbon dioxide out of the air and turned it into a solid, carbohydrates, and it built the tree from those solid carbohydrates. I think that's crazy. All right, the other question is sort of the reverse process. Where does the mass of a tree go if it is burned to the ground and only ashes are left? For example, we'll use a much smaller tree in this case. If a 300 kilogram tree burns down and only 10 kilograms of ash remains behind, where did the other 290 kilograms go? Into the air, back into the air as smoke, as carbon dioxide. When anything burns, carbon dioxide is released, so it's going back into the air. Isn't that amazing how the planet recycles things? Oh my gosh, it's incredible. All right, what do I really need to know, KJ? The term chemoheterotrophic and photoautotrophic. So chemoheterotrophic means you eat, photoautotrophic means you make your own food. So plants make their own food, everybody else eats. Not everybody else, but mostly everybody else. You need to know the equations for photosynthesis and cell respiration and that they are reciprocal processes. You need to know that cyanobacteria are the likely ancestors of chloroplasts and that ancient cyanobacteria were responsible for oxygenating the Earth's atmosphere. You need to know that pigments absorb visible light and that an action spectrum shows the combined effective wavelengths of all leaf pigments for photosynthesis. Guaranteed, you will see an action spectrum on the test that shows the wavelengths and which ones are being absorbed and which ones are not being absorbed. So which light is most effective and which light is least effective. Remember that the red and blue-violet wavelengths are absorbed best, and green and yellow are not absorbed as well. What do you need to know about photosynthesis? It's a redox reaction. Carbon dioxide gains electrons and hydrogens, so it is reduced. An electron transport chain is used to control the release of energy from the electrode. Oh my gosh, let me say it again. An electron transport chain called ETC is used to control the release of energy from the ejected electrons. Oh my gosh, remember the photon comes in, it ejects the electron. The electron transport chain captures that electron and uses its energy. It's going to use that energy to make ATP. All right, moving on. Photosynthesis continued. The light-dependent reactions happen in the membrane of the thylakoid. Photons lead to the ejection of the electrons. The energy from the ejected electrons is harnessed in the electron transport chain to produce ATP. Remember that analogy where the turbine was turning and making ATP? Hydrogen ions are pumped into the intermembrane space. That's called a proton gradient, an electrochemical gradient, or a proton motive force. So many things that they can call it. The hydrogen ions then, through facilitated diffusion, diffuse out through ATP synthase, that pink turbine in the thylakoid membrane, and that's what creates ATP. That's what puts ADP and ATP together. NADP plus is reduced when it gains the electrons. People are going to come in just a second. Water is split to replace the missing electrons, and hydrogen ions and oxygen are produced. In the light-independent reactions, they happen in the stroma. Carbon dioxide is removed by the atmosphere by the enzyme called rubisco. You should know that name. It is combined with other carbon-containing molecules to eventually produce carbohydrates like glucose. ATP is hydrolyzed to power the process, and NADPH drops off its electrons and hydrogens, reducing the carbon molecules. Whew, that's it, ladies and gentlemen. Have a great rest of your day.